All right, welcome. For those out in the foyer, Tom, I know that you'll be allowing them to kind of walk in, so thank you. Okay, everyone, we are so excited to have you all here. Thank you so much for coming, and welcome to the first annual Exchange Forum, a Patient Innovator Forum. My name is Nicole Boice. I'm the founder of RareX, and just honored to have you all here. Get super jazzed about all of this, so we're very excited. So a couple of words of thanks. So a big, huge thank you to Global Genes for allowing us to sneak in on the back end of their event. The flowers are from Global Genes, the staging, so we're super, super grateful and excited to be able to partner with them, and just, you know, gratitude for their generosity. A special thank you to the sponsors, because none of this would be possible without you. Horizon, California Institute for Regenerative Medicine, Kazi, Alexion and Vitae, the Assistance Fund, and Lorem and RTW Foundation. So thank you so much. The last bit of thanks not only goes out to the amazing Global Genes team, but also to our board members, because we have an amazing group that have descended upon San Diego to join us today. And so thank you for your leadership and for your contributions. I'm only allowed to talk for like 2 more minutes now, and everyone that knows me knows that that's practically impossible, but I promised this amazing panel that they would get as much time as possible. So I will say one thing. The purpose of this event is you, patient advocates, and those in the room, have made a decision to be active participants in driving research. And as a result, you've recognized your role and the importance of patient-level data in supporting the critical research that we know is so important, and to provide what biopharma researchers need to help accelerate future therapies. So, our hope and intention for the next day is that we create a dynamic interchange or exchange of ideas, innovations, insights that you can take forward, and on Friday, when you end up starting your day, you're overwhelmed with all sorts of possibility, action items, and next steps. We want your time here to be super valuable. We know that we don't have a lot of extra time, and so we hope that you can walk away with some really meaningful next steps to help support you in all of your research-driving agenda. So without further ado, thank you, welcome, and we're gonna turn it over to the amazing first, oh, I forgot, there's a couple housekeeping items, sorry. Bathrooms, down straight, if you walk out here to the left, there's a hallway, that's where you can go, take a bio break. Today and tomorrow, we have a chock-full agenda. So we're gonna ask everyone to be responsible for going when you need to go, right? We do not have breaks. And the whole purpose is because we wanted to make sure that every single second of time that you had real quality conversations, discussion, speakers, and so we wanted to make this time super valuable for you. The other housekeeping item tonight, following this event, we'll walk out. There's going to be a combined reception with Global Genes, and thank you to Horizon for sponsoring this. And then we are going to be back here in the morning, crack it on at 7 a.m., and we're going hard until 1. So we expect you all to be here, bright and cheery. We'll have lots of caffeine, and then we're going to hit it. So thank you. I'm turning it over to Morrie Ruffin, an extraordinary board member, friend, partner from Adjuvant Partners, and also a RareX board member, and like I said, longtime friend of Global Genes, the org, and personally. So thank you, and I'll let you introduce your amazing panel.
Thanks. Thank you, Nicole. Thanks for that wonderful introduction. Good afternoon, good evening, everybody. We're really excited to be here today. Nicole came to me a little while ago and said, you know, we're going to be doing this first ever exchange forum, and we really want to kick it off with an inspiring discussion about what we're all doing. I mean, what is this great journey that we're all on here together in trying to figure out how to apply some incredible technology, which you're going to hear about, represented by some of the people here on the stage, to rare disease, and to figure out, you know, to understand what is the state of this sector right now, And we're going to talk a little bit about that. What is the future, potentially? And what are the tools that are being developed by a lot of the companies in the AI community that can be, and the machine learning sector as well, that can be applied here. And so we've assembled really a very, I think, inspiring panel here today, and I'm going to introduce each of the members of the panel here in a second. I just wanted to say a little bit about my background and my relationship to Nicole and RareX. So I am one of the co-founders of the Alliance for Regenerative Medicine, which we started in 2009, which is the global organization representing the cell and gene therapy community. It was really a novel kind of structure to bring that community together and advocate on behalf of a variety of different stakeholders. And based on the success of the Alliance for Regenerative Medicine, we started a conversation with a number of companies in the AI sector in 2018, 2019. This is just as a number of these companies were being funded, created by venture capital and spun out of institutions and really beginning the incredible work of applying these tools to drug discovery. Not that these are tools that hadn't been used before, but there's really, I think, an acceleration, and you're going to hear about that in this work, in the technology that's being used, in the insights that we're gaining from this. But it needed an organization, and so we essentially borrowed the model that was the Alliance for Regenerative Medicine and created the Alliance for Artificial Intelligence in Healthcare and launched that in 2019. And I say this because it's really these organizations that need to be partnering with groups like RareX. And these are the companies, the individuals who I think can bring a lot of the tools and the insights and the experience to the table to partner with the rare disease community, and especially around the incredible work that RareX is doing in bringing these disparate data sets together in platforms and models that are usable and accessible to the broader community. So we're really excited to talk about that, and I think today we're really going to be talking about the potential of that. So without further ado, we're going to start with introductions. So what I'm going to do is ask each of the panelists to introduce themselves, since the topic of this panel is to be thinking about big data and data in a slightly different context than perhaps we've been used to before, but really how it can be applied to drug discovery and how it is being applied today. So I'm going to ask each of them to do that, and I'm going to start with Ryan. So Ryan is our patient representative on the panel. He's also an innovator and thinks a lot about this. So maybe Ryan, if you could introduce yourself, and then we'll go around to the rest of the panel.
Yeah, I'm Ryan. I've got some typos like everybody else here. A couple of mine happen to be on my GAA gene on chromosome 17, which codes for Pompe disease. I'm a naturally curious person. My professional background it has been in motorsport, airplanes, rockets, and satellites. And so I have always found empowerment through knowledge and its pursuit. It's brought the same thing with my diagnosis, and I've been trying to chase down sort of comfort, confidence, acceptance, all those kinds of things in the same way. I find that I'm more curious and have more questions and generate questions at a faster rate than I can find answers, and some of the answers didn't quite fit and make sense to me. So my interest in this kind of space is taking a look at what data we do have, what data should be reasonable to get to reset our understanding of Pompe disease, and then what we're learning about along that journey that can be translated to other diseases and resetting understanding there.
Great. And then so to Ryan's right is Bob Deans. Bob is the Chief Scientific Officer of Synthego, and he's someone I've known for the better part of, I think, 20 years.
Yeah, we'll keep that
Yeah, we'll keep that discreet. But Bob really does bring a unique skill set to this. I mean, this is someone who really understands technology across a variety of different technology disciplines, and in many ways, that's what Synthego represents. It's the synthesis of a lot of these platforms, and certainly relevant to what we're trying to do in rare disease. So,
yeah, thanks, Morrie. So I am Bob Deans, and I am CSO at Synthego. Synthego is a genome engineering company, and we actually have some overlaps with Ryan. The Synthego was founded by two SpaceX engineers who came out and looked at the biology space and realized that engineering principles could greatly reduce the dimensionality and complexity of disease modeling and therapeutic development. So it's incredibly rewarding and attractive to me to shift out of my background in cell and gene therapeutics and move into a platform position where we could start to really deliver this class of new medicines and overcome some of the regulatory roadblocks, some of the access problems that exist. So it's been an incredibly rewarding environment for me. We use AI or machine learning and big data in a couple of ways. The company actually produces GMP or clinical grade therapeutic products, and we optimize those in terms of our process development and our purification processes, our synthesis. And we always use iterative loops of machine learning to understand our variants, reduce our turnaround time, really build things out around the quality component. I think the more interesting way that we use big data is in disease modeling, and we do that to validate targets that are suggested by genetics and put them into a test bed, a cell type, that can give us a link of the gene difference to the biology, the functional output. And so we do that using induced pluripotent stem cells, iPSC. And so it gives us a test bed where we can start from a fully undifferentiated cell and have a cell replicate its developmental pattern. So we can go from a cell with no mature purpose or function and turn it into a neuron or a cardiomyocyte or another cell type. The reason that's valuable, I'm going to take a couple of minutes.
No, this is fine.
Yeah, the reason that's valuable is it lets you study the consequence of a genetic difference in terms of epigenetics and control of the developmental process. And so many disease pathologies are only seen late in maturation or late in the body, but they are a consequence of genome regulatory changes that occur very early in development. So it gives us a link to look at not just the patient or the individual at an adult stage, but it lets us look at the process by which the tissue's developed, and we can look at some gene targets very effectively in that way. So I should probably stop.
We're going to come back to that. So
Yeah,
that was great. That was a good teaser and appetizer.
Yeah, I'm really motivated around patient access and solving these problems, and you'll hear a lot of fervor and conviction to help you guys get where we need to be.
Right.
Kazi.
So next up is Kazi Mustaqhusain, and you go by Kazi, right? Everybody calls you Kazi. Okay, good. So we're delighted to have Kazi here. He is the global digital strategy leader, and there's, I think, about 2 or 3 other titles that go along with that at Roche. And he's somebody who thinks about this in depth every day. And we're really delighted he could be here to share his experiences and also give us the perspective of a firm like Roche, who is collaborating across the board with companies in this space and also with patient organizations and patient groups to really kind of think about the entire enterprise. So if you could introduce yourself.
Yeah, so I'm part of Roche Genentech. I joined Genentech when it was almost like 4,500 employees. And then we had like 2 of the big drugs approved, or 3 of the drugs approved. Avastin was one of them, which was like, one of the personalized cancer therapies. And then since then, I've been part of Roche, after the merger and, had a global role now for almost like 5 years. And it was on personalized healthcare informatics. And now, since October, I'm taking this role of the global strategy leader. So as part of PhD, I've done pretty much everything like you could think of from an infrastructure perspective. So building, foundational technology platforms on, Amazon, Google, Microsoft, laying down high-speed network, connecting with Giant and Internet 2, to all the way building, data analytics platform. And so we deal with like all types of data, starting from clinical trials data, EMR data, claims data, different type of clinical imaging data, genomics data. And so I think, when Nicole and I first talked, I think this was like a year back, right, Nicole? This was like my, honestly, first foray into rare X, because before that, I was like totally focused on oncology, on, non-rare diseases. And I think like, with the discussions with Nicole, I think, there is a lot of techniques that we can apply from the mainstream data analytics learnings into the rare disease. And I think some of those can translate. Some of those we have to very much specify and be much more targeted for rare X community. So I'm very excited to be here.
Fantastic. All right, and last but not least, to Kazi's right, we have Stacie Calad-Thompson, and Stacie is the Chief Strategy Officer. There's another title there too, or head of drug discovery.
Yeah.
Okay, good, I got it. For BioSymetrics. So this is a new company, and it's been around for a couple of years now.
Yep.
Okay. And I thought it was based in San Diego, but it's not. But you can describe its origins and where it is. So please introduce yourself.
Sure. Hi, I'm Stacie Calad-Thompson, Chief Strategy and Officer and Head of Drug Discovery at BioSymetrics. We are a phenomics-driven drug discovery company, a startup that uses machine learning to integrate and link and navigate clinical genomic data sets to arrive at insights, biomarkers, genes, druggable targets. And then we use that data and translate it through our knowledge graph to appropriate model systems where we can experimentally test and validate and then pursue drug discovery programs. So we use machine learning along that entire kind of life cycle of drug discovery. I met Morrie initially as a founding of the AAIH at the time I was at GSK, and I've been working in applications of AI and machine learning since about 2016 when GSK was investing in this heavily, and one of the first roles we had there was one of the first roles I had was the establishment of the ATOM Consortia, which was a public-private partnership set up to build open access tools for AI and machine learning. And so looking forward to talking about more applications of data and learning from that data to accelerate drug discovery.
Fantastic, thanks. So so the way we're going to run the panel today is we have a few discussion questions we're going to pursue as a group. We want to leave time for questions from the audience, so I'm going to make sure we have, so Nicole, what do we have?
There's a big clock in front of us.
26.
Okay, so we'll make sure we leave 10 or 15 minutes at the end. If you have a burning question, please save it. We want to make sure we get to questions from the audience. But this is a big topic, and there's a lot of ground to cover here. I think we're going to start with just making sure we have a firm understanding of what we mean by big data, but not necessarily the term big data. It's really how data is used by these companies. And so I'm going to ask each of the panelists to kind of give their own definition of it in the context of how they're using it. But Ryan has got some really important insights here because he thinks again about this on a pretty regular basis. So maybe if you could kick things off and then we'll go to the other panelists.
Yeah, I think just the way the discussion started is that, in rare disease, we're often told, oh, there's not enough data, or there's not enough people, or there's not enough. And so aspirationally, when we talk about big data in rare disease, it's just trying to figure out how we can take these real-world problems and translate them into questions and study and things that we can study and multiply those data sets by orders of magnitude so that we can have a lot more powered study and not just accept these sort of answers or opinions or sort of facts about our diseases that are not actually facts. They're long-held beliefs that are based on really underpowered studies or knowledge. And so how do we replace that with something new? So one of the ways we're doing that, I'm doing that is from the start is we've got newborn screening in Pompe disease now, and that's a direct measurement of incidence, and instead we've been using incidence from 3,000 blood spot cards 20 years ago as the current figure. So we replaced that. We've went out and got 11.6 million newborns screened from around the world, reestablishing incidence and doing it along the way, coming up with a path to unlock publicly available genetic data sets, hopefully to provide other rare diseases sort of a decades forward jump into what their incidence and basic epi stuff looks like so that we can go out and build business cases and build models and get samples and participants for these
Who is
folks.
leading that effort, though, that you're describing? I mean, how is that, I mean, obviously you're invested in this, but I mean, can you just
I
describe
am.
how that's happening?
Yeah, no, I've done all that sort of in my free time.
Oh, okay, all right. That wasn't a setup. All right, Bob, maybe if you could go next.
I already started on my why I love big data. So I think, and what I'm reflecting on here is experiences we have with partners or academic collaborators, so I really can't name certain disease alleles or talk to specific partners, specific stuff. But what we've really been anxious to do is to explore the utility of the iPSC, this non-differentiated pluripotent stem cell, to give us predictors of response to disease. In other words, can we, when somebody is diagnosed or has an allelic difference, can we then take a broader look at his genetic background, their genetic background, and make predictions on whether they'll respond to current treatment or not? So what we're going towards is that once you've identified a driver gene that's associated with disease, you see individuals that are able to either show you accelerated pathology or appear to be less susceptible in representing pathology, the penetrance of disease. So that's happening because there are secondary genes or proteins that interact with that driver, sort of the rheostat that is able to compensate or exacerbate the consequence of that allelic change. And so those secondary drivers can be identified by using big data in terms of transcriptomics or other proteomic approaches to characterize the cell. So imagine you have a test bed with a normal genetic background, and now you understand that there's an allele in Pompe or otherwise that you can drop into that background. Now that you have the mutation in that cell type, you can ask, how does the function of the cell differ compared to the normal state? And what you're able to reveal by using these big data sets are the differences in these secondary drivers or the associated biology within the cell that dampens or accelerates disease. So what it gives you is a cascade of genes that serve as diagnostic markers associated with the specific patient response. And Stacie's going to talk a lot more about this. What we've done is looked at patient-specific iPSC. So now we're studying the disease allele, the individual allele, in the setting of the patient background. And so now we can start to make a comparison. How does the mutation in the healthy cell differ from the mutation in the individual's population with respect to all these secondary drivers, the bioinformatic-driven analytics that occur? So you can clearly see that when we start to stack up a number of individual-specific iPSC like that, we start to tune in on all the variances that exist that are relevant to the individual patient. So the reason we get a lot of interest in that from partners is it allows people to stratify people for their likely response to clinical approach. Because what we're accessing in combination to the omics data that I'm describing are the clinical and wellness data that's collected in the community, and we can test rigorously mathematically for correlation. So it's hugely valuable to understand and not put people on inappropriate regimens, or at least be able to shift over in a personalized medicine approach, look much more specifically at a rational approach for disease moderation. So for us, it's been an incredibly powerful tool just for unlocking the ability to stratify and understand disease. What it also does for us is tell us that we can take those differences and go into either a gene-based therapeutic approach or a small molecule-based therapeutic approach. So one of the things we had done a couple of years ago in terms of response to COVID is work with an investigator that was studying how COVID proteins, when they infect the cell, interact with human proteins. And so was signing up sort of the structural counterparts that exist between the viral genome and the human genome. So our idea was that we could deliberately knock out each of the human genes that was interacting with the COVID protein and test whether or not that was necessary for viral replication. So what we're doing is then setting up candidates that represent interaction points between the COVID protein and the human protein that's suitable for drug-based structure-function-based drug discovery. So one of the reasons that gene editing has been so attractive in this space is we can do things very quickly and generate data very quickly. So it's not just big data, it's also rapid data accumulation. So what we did, one person in our group sat down and ordered 1,000 different guides. That's a guide is the small RNA that would target the gene and result in knockout. We took 3 different cell lines and made 300 different gene knockout pools, shipped those to investigators in Europe and New York. All of this took place in 14 days, and we had 12 candidates that dropped COVID infectivity by more than 2 orders of magnitude. We're down in the single % digit range. Out of that, 2 of those have moved forward into registrational studies in treating COVID and reducing complications of disease in a 10-month period. So we were able to do that because we join up with people that are able to do, like now, in silico modeling of how protein-protein interactions take place, and you can use that big data set to screen against drugs that the FDA has already approved for purpose in other disease settings. So what we were basically doing is capturing the diversity and information around these structure-function elements, human and COVID proteins, matching them up by in silico predictions with already approved drugs, and ran with it. So this is the kind of thing you can do as you start to hone in with larger data sets and experience in a certain disease setting in terms of getting a very rational and rapid therapeutic design in play. It's an incredibly powerful.
That's a great example. I want to come back to that again. I mean, as we talk about applications in this space.
Yeah.
Again, and so that was a great example, I think, to set that up. Kazi, if you could, again, talk about this again in the context of how Roche works with data. I mean, and you described kind of a process and ecosystem. So maybe, if you could explain that a little bit to us and why you think this has great potential for the rare disease community.
Yeah, so we actually started with the term big data in, I think when it started like 2013-14 timeframe. And then we realized that you need actually statistically significant amount of data. So it's not that you need like hundreds of thousands records. It's more, what are the hypotheses that you have and what are the questions you're asking? And you have to have the data based on that, right? So we coined a term internally called MDAS, meaningful data at scale. And basically what it means is, like, let's say if you take oncology. So oncology, we look at like 4 modalities in terms of like really looking at the patient, disease progression, right? So one of it is obviously clinical trials data or EHR data. So it gives you like, the output from there. Then we look at imaging data a lot in terms of like CT scans. In particular, we look at genomics data quite a bit, and then we added the patient-reported outcomes because if you want to measure the quality of life, you can only get it by the PROs. That's what we found, right? So that's kind of a combination that we have for oncology type of data sets. And then if you go into like something like multiple sclerosis, then we use a combination of obviously like clinical or EHR data, and then we use a lot of MRI scans of the brains, and then also we use the digital health apps that we give to the patients that can actually measure MS progression, like every single day, whether they are going to the doctor or not, right? And then for ophthalmology, we use a lot of eye images. So we use like OCT and color fundus and fundus images like out of that, right? So depending on the type of questions we have, the type of disease areas we look at, we look at data differently. So from the rare disease perspective, that's where, with RareX, we are focusing on degenerative neuromuscular diseases, et cetera. And then we are looking at, what are those key modules that we need to build, right? So is it around, the sleep patterns? Is it around like quality of life? And we are trying to build those measurements using mostly like PROs right now.
Fantastic. Great. Stacie?
So we've got 2 categories of data that we think about. There's patient data, electronic medical record data, and I liked what you said when you mentioned meaningful data, because it really is about signal to noise. We've worked with as little as 50 patient records and as much as tens of millions of patient records. Each use case is different, and we use that to get to phenogroups of subsetted patients so that we can better stratify and segment and understand patient disease and the biomarkers that affect subtypes of diseases. The second set of data that we've got is in our experimental data, and maybe there's a third set of data, which is like publicly available data as well, because we've done a lot to integrate that into our phenograph, which we use to kind of navigate between the clinical insights we discover and the experimental models that we choose to explore. So we're building out automation capabilities, and similar to what Bob had said, our model of choice right now is zebrafish. We have an ambition to one day go into iPSC cells as well. But zebrafish are great for some of the disease areas we're in, which are cardiometabolic, neurological, and rare diseases. You can genetically modify them, and from guide injection to phenotype observed is 48 hours. That means that if you scale that and can screen, hundreds of genes, you're able to get lots of combinations and subsets so that you can understand better genetic backgrounds and link the phenotypes that you observe to the clinical phenotypes of a patient. We've also got then capacity to screen, tens of thousands of compounds against those different backgrounds. And that gives us a starting point to really accelerate drug discovery. All of that data then gets fed back into our knowledge graph as well, so that as we're building over time, we've got more of a knowledge base to connect those clinical data with the appropriate models that will translate into drug discoveries that work for the patients that we subtype to begin with.
So maybe we should, I want to kind of extend this into a discussion about what does this mean in terms of actual therapeutic development? I mean, obviously, this is the background for drug discovery, but at the end of the day, we have to get drugs to patients, and we have to do it faster. And in many cases, we have to just do it because it's not happening, especially in the rare disease community. So, I know there's a lot there. I mean, obviously, you can't tackle every single disease, so you have to make choices along the way, and we have to demonstrate that we can do this. One of the things that is interesting about what Synthego brings to this is they think about it with an engineering mindset, and how do we really do this at scale and make this something that can move the needle in terms of the process of developing new drugs. So maybe, and Ryan, if you just indulge me here for a second as I go through this. I want to ask the question, it's not really fair of Roche, but a lot of times, I know we have Walt sitting in here, VCs will, in initial discussion with companies say, tell us what makes you unique. Why is what you're doing really relevant to what we're trying to accomplish here? So I think you've done that, I mean, as you were answering the question about big data, but as it pertains to actually developing therapies, I was wondering if you could each take a swing at that. And I'd love to hear how Roche responds to that too, but maybe, I don't know, Stacie, if you want to start, and you can, talk about partnerships and who you would be working with as well.
Sure. I think our three differentiators that I list off are access to clinical data, which we've got 77 million EMRs right now, but are always looking to partner with new data partners. We've got machine learning capabilities in our phenograph, as I described, that really link human biology to those of model systems. And then our kind of automated phenotypic platform that enables us to screen rapidly. And in talking about therapeutics, two of the programs I can highlight really quickly. We've got one in epilepsy right now where we worked with a partner. On that case, we only had 50 patients that had Dravet syndrome. We're still recruiting additional patients, but we're able to get insights from that that enabled us to design the genetic backgrounds for the zebrafish that then we screened compounds against. And that is an example where, because we were using a behavioral assay and observing seizure-like behavior in zebrafish, we were able to get really detailed insights that something like that's much different than just a cellular assay would have given you. The other example I'll talk about is familial dilated cardiomyopathy, which is a genetic inherited cardiomyopathy This is one where there are some known genetic backgrounds. We used our phenograph to prioritize which targets we wanted to go after, and then have kind of a subset of control genes as well as novel genes that we're exploring. And with our computer vision capabilities, we're able to observe in a zebrafish ventricles that are bigger and ejection fraction that is reduced. And that's, again, an advantage of going into an in vivo system rapidly. Then we can screen small molecules against those assays that we've developed. And again, it's all about speed to doing that. So being able to predict and sort of do a virtual phenotypic screen first, that then it better informs and makes your experimental work more efficient. And being able to capture all of that data and learn from it is what's going to speed up drug discovery.
And just one last quick question related to that. So how far then will you take it? I mean, so if you find compounds that you like and you want to take it to the next, is that, I mean, do you do that with a partner or?
We're looking to partner early as
Okay,
a
so,
startup.
all
We
right.
don't have further capabilities yet. So at this point, yes, looking to partner early.
Maybe talk to the guy next year. But anyway. So Kazi, could you describe that? I mean, and maybe also in the context of kind of what you're looking for in terms of capabilities, and that you bring in. You either build them in-house or you access them through partnerships.
Yeah, maybe like I'll start with the data categories. I think, going back to Stacey's point, so I think we look at data in 3 categories. So one is, the clinical trials data. So we have a lot of data coming from our own clinical trials that we can use for secondary purposes. The second is the real world data. So that's where we look at partnerships with, external providers. So it could be with, academic institutions. We have Flatiron as one of our companies, but we still have a firewall between pharma and Flatiron in those regards. So we have to buy the data. So we buy a lot of data from external parties, and it's highly curated data, and it could be EMR, imaging, all of those different categories. And the third one is the public data sets that we have access to, right? So we use all 3 and generate this meaningful data at scale. So that's one. In terms of line of business, so we are in 2 main areas. So one is developing diagnostics. So, the COVID testing was one of the things that, Roche did as part of the diagnostics, for example. And then the second part is the pharma and the life sciences part, right? So a lot of data we use is to actually create this early and accurate diagnosis. So either like the testing that goes to the market. And I think, if you think of rare disease patients, diagnosis is one of the key issues that you have because, you go to a physician and they have not seen certain type of characteristics in the patients, right? So giving them the right tools, I think would really help, accelerate the journey in terms of the patient. And then when you go into therapy side, I think, what Bob alluded was developing therapies that are, in silica, what I call, right? So before we would have wet labs and we would go through the whole process. Now we use a lot more in silica and then, use the wet lab for the, testing that hypothesis out, right? And then as you move into the cycle of clinical trials, one of the biggest issues and the huge cost is around clinical trial operations. So that's where we use, first of all, the inclusion criteria of which patients will benefit from which therapies. So that's one of the key things, and that helps the trials be much faster, also more cost effective, right? And then when we go and the drug is out in the market, we want to do continuous monitoring. And this is where real world data, PROs become very, very helpful, right? So that's kind of the whole cycle. So it's going from early diagnosis to drug discovery to clinical trials and faster clinical trials to basically ongoing monitoring of the the disease in the patients. And based on that, then we look at quality of life measurements and see how we can make it much better.
Okay. So Ryan, before I come back to Bob on this, because I do want to hear the Synthego response. I mean, as you're thinking about this, I mean, you obviously have a lot of peers in the community. You are, I think, obviously talking to people affiliated with companies or efforts to start the process. I mean, as you listen to kind of some of the responses from Bob and Kazi and Stacie, do you have confidence that we're moving in the right direction as we're using data? Or what do you think is missing at this stage?
Yeah, so I have full confidence, and I think there's a lot of really incredible tech. So I think I have confidence in that, but not necessarily the way that it's being applied. I think that there's a lot of stuff that we've learned from manufacturing, and that's maybe a bias because that's my background, and operations, that when we look at the sort of what fast means when you're talking about fast timescales or what this all looks like, it doesn't actually
Right.
doesn't actually hold weight that we have our aspiration and our bar set high enough. And when we look at the consequence of that, it's lives and livelihood at it. So what I'd really love to see is figuring out how we can work with data to accelerate knowledge in a truly aspirational way, not just like, shave a year off of trials, but shave decades off of advancements. And I think one of the ways that data plays a role in that is we're used to answering these kinds of questions. We have a data set that is this, or we go out and buy a data set that is that. And that's maybe one of the things that RareX is trying to do in terms of shaping the space differently, is we're not looking at this as we have ownership of subjects' data. We have people who we're trying to help and we're trying to be of service to, and they can participate, and we can sort of mutually benefit from that. Them in lives and livelihoods, us in doing really cool science, and then there's plenty of good money to be made on there as well. So I think that's really where a lot of the opportunity is, like taking a little bit more of a high-level critical look. Where is our opportunity? When I talked about our project, why we're focusing on incidents, which just seems like so boring compared to really the cool tech and applications here, is just like what that ends up translating to is we've got about a 5% diagnostic yield in the current clinical model of diagnosing Pompe. And if we were able to increase that to any acceptable level, how much cooler and sort of cooler work could we enable Stacie to do with data sets that are looking at phenotypic differentiation when you have a data set that is 100 times bigger, or give Bob some different samples to play with, so you have not just a single individual allele, but then now thousands of different alleles that you can go and play with, and how much faster could we learn, and then get Kazi to write a big check to translate that into.
Is that what you do? You write big checks, Kazi?
I think I want to just piggyback on that. Sorry, Bob. I think one of the things we are looking at much more seriously is real-world evidence. So one is, you go through the trial process, and it's like, multi-year process, very expensive, right? And especially in rare disease areas, and even like in oncology, right? Using real-world evidence, for clinical development, but also like from a regulatory perspective. I think that's something would be very beneficial as we go forward, because then you can really do rapid testing of things on the market, as we call it, right? And that's an area that I feel will develop much more in the next 5 years.
Yeah, the ability to differentiate amongst the learning, so have better granularity in our feedback loop that will then create higher power in our studies. And I think a lot of times we're talking in this space only about confidence intervals or confidence, and we're forgetting the beta error, and that's really just as important in having a real understanding of what's happening.
Yeah. So, go ahead, Bob.
I just want to reinforce your point about challenging the 10-year development cycle. And I think a lot of that is the naivete or the position the FDA is in, the regulatory agency, to look at the N of one patient as an individual. And we're not amalgamating the treatment experience that's gone across time. And so I think the way to shorten this and to get some of the new medicines, particularly gene-based, in is to work aggressively with the agency about the regulatory requirements and the development costs.
Yeah, and that's absolutely, that's part of that collaborative model. Like, this isn't my data, this isn't this. We're partnering with the patient communities, partnering with regulatory bodies, and we're kind of like saying, okay, how does this all look if we look at it differently, if we're all playing with the same set of rules on the same pitch? Now innovation really does win.
Yeah, so the key is finding a lever that forces the therapeutic sponsors to drop a majority of their clinical response data into a public data set. And I think the first attempts I've seen at doing that have been to try to acquire some of the placebo and natural history data into a common pool. What it does, allows people that are trying to develop the therapeutics to have access to big data so they can do appropriate design. And so, the lever has to come from the academic institutions or groups like this, because the therapeutic sponsors limit themselves to their fiduciary responsibility of driving profit. So somehow that model's got to get broken so we can access the experience data and build these things out aggressively, independently. So I jumped in.
No.
Yeah.
I think I want to just cover one or two other things, and then we've got till the bottom. Okay, but do we have till the bottom of the hour? Okay, all right. So just one, yeah, and then I want to open it up to the audience. But, so one of the things we talked about as we were prepping for the panel was kind of where we are today with these tools and the technology we have at our disposal. And you've heard the panelists talking about that and really, kind of thinking about where we would like to go with this. Is it a data problem, as Ryan was describing, or is it a technology problem? I mean, obviously, it's iterative as we continue to improve the tools that we have with the data sets that we're using. But I'm curious, because Stacie, is familiar with a lot of these. I mean, do you feel like the AI and machine learning tools that we have are maturing at the same rate that we're accessing the data, or not, or are we catching up?
Yeah, I think there's lots of opportunities where where there is convergence happening and maturity happening. The data and access to data is still, a barrier, I think, as we start to collect data in different ways, especially, patient data right now is collected by doctors and coded according to insurance reimbursement codes and not in a way that enables research to happen.
Right.
I think those are starting to change, especially through groups that are focused on enabling research to happen. So as that comes about and knowledge bases continue to grow and machine learning capabilities grow and the kind of automation and technology to integrate drug discovery capabilities, it's the merger of all of those together that I think does have the point where, in the future, someone with a rare disease can show up and give a blood or a tissue sample and be able to have it sequenced rapidly and be able to get their phenotypes put into something that then translates it out and says, actually, we're going to use your cells to build out an organoid model, and we're going to screen these 5 to 25,000 drugs or something against it, and this is the right drug combination for you. Like, that's going to be amazing.
Yes.
Yeah, I'd agree. I think we're data limited or data starved right now. The tools are further along the runway, but this is a problem that we need to build the machine that allows us to iterate very quickly, and that is fed by data.
But related to that, are we modeling disease in a static way, or are we able to really understand disease progression to using these tools? I think that's part of the process. But are we at that stage yet? Do we really understand that?
I think, again, access to longitudinal patient data is still like the limiting factor there.
Yeah, I feel like there are maybe 4 parts to this problem. So I think one is definitely technology. And I think, to large degree, the cloud, I think, has solved quite a bit of that, because in the past, data would be stuck in data centers and very hard to get out, I think the second one is this data pooling. So as you said, I think still, I don't think we are there yet in terms of having that longitudinal, and not just longitudinal and highly curated data. That's something that you can run models on, right? The third one is the AI techniques. And for us, we partner with a lot of institutions to really advance some of the techniques. Because if you look at real-world data, it has gaps in it compared to clinical trials data. Clinical trials data, you can get exactly all the different fields. In real world, it's very hard to get that. So there are techniques that would actually allow you to uncover in that. There are also techniques in terms of allows us to do, and we talked about this, is federated data. So if data stays in different places, how do you then run models, right, across these data sets, and seems as if it's one giant pool, right? And I think there are new techniques being developed there. And I think the last one is the regulatory part. So even if we do all those other things, if we don't have the right regulatory process in place, and especially when we go to cell and gene, it's a totally new area. It's completely new area. So
we have
you need 2 things. One, the regulatory teams need to really understand what that is, and second is the capacity. Because if you look, I think there's a projection that by 2030, there'll be more than 60 cell and gene therapies approved in the US, right? That's a lot of work for the FDA in addition to what they have, right? And so if you don't have the capacity to do it, as well as if they don't have the technology know-how, I think it'll be like very hard. So to me, it's like, all the 4 things: technology, data, AI, ML, and then regulatory.
Let's jump to the audience. I mean, unless Bob, did you have a last word?
I'm good.
Okay. So I think we have a couple mics here. So raise your hand if you have a question for the panel here. Is there somebody out there? We can't.
So I'll kind of tee it up. So what I think is so amazing at this time in the ecosystem and kind of, over the last several decades, like everything looks so different today than it did even 7, 8 years ago. The fact that we have this group on stage talking to patient advocates, talk about opportunities with organizations like them, that you can and have the ability to partner with is pretty extraordinary. And so again, the hope for the next couple days is really, or the next couple days, the next day is really, really sorting through and figuring out what that collaboration and partnership opportunities look like. And so having said that, I guess, I just was teeing this up, but so is there any thoughts, questions? Oh, perfect.
Did you want to turn that into a question? I love the comment, Nicole, if you want to turn it into a question.
Sorry.
So if this is not related, so if we do want to turn that into a question, we can.
Go ahead.
Okay, great. I just wanted to get some impression on, we talked a lot about predictability and using the tools that we have to try to let the data tell us what's going to work before we try it in vivo. And so, Stacie, in particular, in your situation with the zebrafish models, what can the data do to advance past that and predict what's going to be translatable if it does work? What's going to work in the next mouse model, or what's going to be like effective in other things, because the genetic homologs aren't always one-to-one.
Yep. One of our core pieces of technology we call the phenograph, and it's a knowledge graph that links human phenotypes, human genes, human diseases to those of model systems. So we do have, human gene to zebrafish orthologs mapped, but we've also got phenologs mapped, where we have observed this phenotype in zebrafish and been able to connect it back to a human disease phenotype. So there's multiple dimensions here by which we've got increased confidence in translation. It helps in diseases where we do have some knowledge of translation because things have gotten far. We can then backtrack and generate data that builds that confidence. But it's also about active learning, I think, as well. So being able to test many different things, feed that information back into the models to improve probability of translation as well. And it's not all about one model. Zebrafish is a useful model, but it is not 100% as well. So we know its limitations in that, and that's where we go into cellular assays and things like that.
You also raise a good point that you don't have to hit 100% to be successful. You're really looking for dropping down to first principles, what are the 1 or 2 variables that are disease-linked, and when you cast in zebrafish, can you get a hit? You can capture 80% of the opportunity. You can really move directly. So it's really about increasing data accumulation and insights as you move along that path.
Yeah.
So I'm really attracted to the models that you guys have. I like
your models too.
Maybe
we can share some data.
You got to realize what's being built up here.
Because our phenograph does have, cellular predictions in it as well. It's got mouse models, so that when a mouse model will be translatable and when it probably hasn't been. So.
And there is a move from the FDA to gather experience data like that and drop the preclinical requirements, which accelerates your process through the regulatory cycle. So gathering that data is just hugely usable. Yeah.
A question related to this is, the projects that you select, depend on a number of different things. So, you all are still fairly early on in your evolution. Is this, what does it ultimately look like? Is this an engine where you can offer it to a variety of different folks who can come in and then you can turn the crank based on what particular disease they're looking at. So the funding for this has to come from somewhere. I mean, are those from, I mean, who are the collaborators that you have in mind as you're thinking about your model?
About our partnering model
Yeah, your
and,
partnering
and
model,
growth
yeah.
model?
I mean, like early, like, I mean, in terms of project selection, and then obviously, I mean, you know, or you can build the programs in-house and then license them out. So I mean, what is the balance?
We're looking for partners that can bring data.
Yeah.
We're looking for partners that have an ability to take some of the targets that we have predicted or believe in the vision of sort of the drug discovery screening and be able to help us invest in and push programs through that pipeline. You know, we've got some proof points to start with, but are happy to generate more proof points and turn them into actual programs.
Great. I think there's a question back here.
I think my question was very much along the lines of some of the comments and questions you all have been asking each other, but I feel like in our heads we have a bunch of knowledge graphs and we're querying each other's knowledge graphs in these conferences like this, and it'd be great if you could, the knowledge graphs and other ways that you've structured your data in your various companies, if you had an API that, you know, I could just query your API, maybe even have like some sandbox of your data so that I can play around a little bit, understand what you have, so that I know what kinds of questions I would even bring to you. I don't know, I'm just brainstorming about how would this actually look if we were going to build an ecosystem where we could share easily with each other, even if we just had one-off queries.
Bring us your one-off queries and we'll do the search. Like, that's an easy way to start the conversation, I think, as well. We've got, we don't have an API, but we've got, you know, an internally accessible one that we can do rapid searches for. We had, you know, a collaborator from a rare disease organization come to us, and we were able to rapidly sort of look at an expanded gene network of things that we could probe or look at, and as we're happy to do those types of things to explore what we could do together.
We did like some POCs in terms of homomorphic encryption. So this is like where it's not APIs, but it's more leaving the data where it is and then running the models across. So we tried to do some POCs and pilots on that. I think it's still, the technology is not there, but you know, there is a lot of research going on in that area, and I feel in the next, like, you know, couple of years, we'll have, like, you know, some significant changes there. And I think that would change the game, because then you could really plug into, like, you know, what you have, what Bob has, and like run queries across, right? So first you can see it, but then also like run queries. What it also does, it hides, you know, who has what, because that's something that sometimes, you know, companies are not, you know, as open about. So it hides like where the data is coming from, but you can get the same results.
Oh, that's interesting. I think there's another question here.
I'm going to go a little bit off script and off the topic, but I think it's a big data question that's in a lot of our communities. So we're all looking for translation and new drugs, but a problem that really haunts me is that in certain fields like epilepsy, there are lots of drugs out there already, and it seems that we're not taking advantage of data that's already there to find the best therapy available now. And these are, you know, you take one drug, you take a cocktail. So these are essentially combinatoric problems. And so I know that it's not incentivized by new drug companies to, you know, fully utilize what's out there. But from a patient perspective, I think there's a lot of gold to be had in what's the best therapy out there right now for these individual, you know, syndromes. And I don't think there's a lot going on right now with getting people to the best that's there right now so that we can improve their lives before the magic therapy comes along. But to me, this is a data problem, and nobody's looking at it, and maybe nobody's incentivized. But I think that there's a need in multiple different modality, you know, areas, behavioral problems, psychiatric problems, all of this, and we're creating more drugs, and if they're targeted, great, but a lot of them are still, pathway, and it has multiple implications across. So how are we capitalizing on this?
I can take a sort of similarly wild response to it. I think this is sort of an overall ecosystem health thing that we're talking about here, where the incentive structure for the ecosystem is not aligned with actually the best health of it or the health of its individuals. So we focus a lot on solutions, and once we think we have enough of an understanding of the problem, we really go after solutions, and because that's where we make our money back on it. There's less incentive on understanding where real money is being spent and where real investment is happening. So that's where sort of we as a community, not necessarily looking at any individual company to say, okay, be a moral leader here and give it all up for free. We as a community can say, hey, how do we build a different model where we work together and highlight what's important for the people that we're serving, which are ultimately the patients, and develop the understanding that we need that will then feed into all these cool other techniques that move us really quickly through these like questionably useful drugs. By doing that, we can also work with regulators, accelerate that whole cycle. So now it changes the incentive when you go from a 10-year development cycle to a year development cycle or whatever aspiration that you want to put on it. The return on investment looks completely different as well, and now we can distribute funds differently throughout the ecosystem than we would only when we have to like the only chance that we have to do it is by investing a lot, having an outlay of cash for 10 years, 20 years, whatever, and then hope that it comes back big in the end, and then we're incentivized to hold on to that thing. And we talk about like longitudinal studies as being a thing, and I think longitudinal studies are great and they have to be a part of the strategy, but they should not be anywhere near the top of the strategy because by definition they're rate limiting for how quickly we can solve problems and to solve problems we should focus on understanding them. So that's an aside.
Nicole? We have 2 more questions. Oh, we did, okay. All right. Go ahead.
This may be a relatively quick question. It's sort of a follow-up to the idea of the phenologues and how that's being cataloged to look at translation between models and humans. And what I'm curious about is the extent to which that is informed by our decades of failures and the large amounts of data that exist of things that didn't work and didn't translate in our very extensive pipeline towards drug development and numerous failures along the way. And whether that data is even accessible, and does that help build in improved chances of success?
That's a great question. In my previous organization at the Atom Consortia, that was one of the goals, was looking at, GSK had donated data on 2 million failed compounds so that we could learn from it. And I think there's still a huge opportunity for pharma sitting on massive data sets there of failed compounds and being able to utilize that. There are efforts, to do federated learning on that type of data and figure out ways to share it. But I think it is still incentives maybe aren't aligned yet.
Those failed drugs are drug candidates for treatment for patients that have different allelic variants in the target protein. So you can now come back in and understand in a structure-function context a major allele that's associated with dominant penetrance in the disease setting. The minor alleles are proteins that have folded differently and may interact with the failed drugs to be therapeutically effective. And you have an accelerated development path because often a lot of safety data has been generated on the compounds that didn't make it to the end. So we can, if we have the data both from the omics and structure function side and the performance data clinically, development data, you can start to get into an accelerated alternative treatment approach.
Is Synthego working on any projects?
Stuff like that, yeah.
So why did it fail?
Pardon?
Why did it fail is super powerful for saying what can we do next?
I think this approach is what we're seeing in the last 3 to 5 years. The tools are just here for people to be able to interrogate the diversity, identify the alleles, and then put them into a test background where they can do that type of testing. So I think this is just what we're going to see flowing forward. Should also say before we close that Gene-based approaches compared to viral-based gene therapy corrections are 10 to 20-fold less expensive to prepare in terms of the GMP process and the reagents and components. And it starts to shift this over to an opportunity for institutions to be able to own the development cycle and prepare the reagents and treat. So you're starting to separate your capability of treatment from a dependency on pharma and biotech, and there's an opportunity for academic centers to be much more forceful in this area.
Yeah.
Great.
There's one last question.
Do we? Yeah, okay. Is it a big, long one, or can we?
We
Are you
can
good?
move on. I can ask individually.
Well, because they're all going to be available after, and I just wonder, is that?
It was going to
okay.
be, the topic was around spatial genomics and how can that play a role in
For
understanding.
God's sake, Dewey.
I know we talked about that. We can get that. We can get that afterwards.
Can you promise to meet him up at the reception after? There you go. Okay.
Kazi, it's you.
All right. All right. Thanks, Nicole. So if the audience could join me in thanking our panel for.
Amazing. So remember that there is the reception after, and everyone will be here and available, and grab them, and then grab their cards, and let's follow up after. So thank you guys so much. Amazing. All right, if we can welcome to the stage our next panelist,